History of microbiology. Ancestors of bacteria were the first life on Earth. The first microbes were observed in 1673. In 1665, Robert Hooke reported that living things were composed of little boxes or cells. This led to the cell theory. The cell theory states all living things consist of cells. All living things come from pre-existing cells. Life continues because of the growth and division of cells, and cells are the smallest units of life, maintaining the properties of life. The first observations. In 1673 to 1723, Antony van Leeuwenhoek described live microorganisms that he observed in teeth scrapings, rainwater, and peppercorn infusions. He didn't know what to call these organisms because they were so microscopic, so he called these microscopic organisms animalcules. He then submitted his findings to the Royal Society of London, where it led to questions being raised. Where did these animalcules originate from? This led to the debate over spontaneous generation. The hypothesis that living things or living organisms arise from non-living matter is called spontaneous generation. It's believed that the organisms are going to spring up from a table, or spring up from a pencil, or spring up from your hand. So what this is saying is that living organisms arise from non-living organisms. So this is according to spontaneous generation. As spontaneous generation says, a vital force forms life. So if you read the chapter, you probably would have figured out by now that the vital force was considered to be air. The alternative hypothesis was that living organisms arise from pre-existing cells, and this is called biogenesis. And Rudolf Virchow, 1858, came up with this alternative hypothesis, which we later accepted. So how did we later accept the hypothesis, the alternative hypothesis of biogenesis? Well, in 1668, Francesco Redi, who was an opponent of spontaneous generation, filled 6 jars with decaying meat. He put the 6 jars with the meat, and then he let it sit, and then he came back and looked at his results. And one of them was unsealed, the other one was sealed, and then one was sealed with gauze. Okay? Of course, we see that nothing grew in either of the 2 jars that were sealed or covered with gauze, but something grew when it was unsealed. So from where did the maggots come from? Well, of course, the maggots came from the fly. And what was the purpose of the sealed jars? Well, the whole purpose of the sealed jars was to keep those flies from being able to lay their maggots directly onto the meat. So this helped confirm the hypothesis biogenesis. In 1745, John Needham was for spontaneous generation. And what he did was he boiled a nutrient broth. Then he took that nutrient broth and he placed it into a sealed flask. Okay? So he boiled it in one thing and then he transferred it to another thing. And then he closed it up. And from there, he had microbial growth. But we all know, from where did these microbes come from? Well, these microbes came from the air. So as he was transferring it from one flask to the other, he could have transferred it through the air, or he could have also had a contaminated flask that wasn't, completely sterile. So that's where microbial growth came from. That's where the microbes came from. So here, of course, we know now what caused it, but then that was one of the things that helped to support the spontaneous generation hypothesis. In 1765, Lazarus Spallanzani repeated Needham's experiment. And he boiled the nutrient solutions in the flask. So he kept them in the flask. He heated them in the flask, and then he sealed it. He never transferred it to another flask. From there, he saw that there was no microbial growth, and this helped support the alternative hypothesis, biogenesis. In 1861, Louis Pasteur demonstrated that microorganisms are present in the air. He was the one that did the experiments disproving spontaneous generation, and it helped lead to the development of aseptic techniques. What he did was he took a nutrient broth and he placed it in a flask. He heated it, but he did not seal it. And then he took another nutrient broth. He placed it in a flask. He heated it, but he didn't seal it. So he kind of took Needham's experiment, and then he took Belazini's experiment, and he kind of put it together. And from what we see is that the one that was open to the air, there was microbial growth. Of course, the one that was sealed, there was no microbial growth. But there was a lot of controversy with this because people was like, well, you sealed it, so you did not allow the vital force, which was air, to come in. So hence, the experiment was null and void. So Pasteur was like, okay, well, let me come up with an S-shaped flask design. And what he did is he did this S-shaped flask to keep the microbes out, but to let the air in. So what he did was he first poured the beef broth into this flask, okay? Then he heated the flask. And then he took the mouth of the S-shape of the flask and he placed it into an S shape. So he formed this beautiful S shape. And the organisms, of course, were able to come in, but they weren't able to get to the broth because what was happening was after a long period of time, the organisms would get trapped right here in the S shape portion. Now, what if I came and I tilted this flask and the broth came in contact with some of the S shape? Then of course I now have presented it to these microorganisms, so things will grow. But when he did this experiment, nothing grew. Okay, he let the air in, and he also, had the flask open, wide open to everybody to, have access to see that there was nothing growing. So this was the experiment that disproved spontaneous generation. The golden age of microbiology was from 1857 to 1914. It was the period of rapid development. It began with Pasteur's work, discoveries including the relationship between microbes, the disease, immunity, and antimicrobial drugs. Pasteur also showed that microbes are responsible for fermentation. Fermentation is the conversion of sugar to alcohol to make beer and wine. Microbial growth is also responsible for the spoilage of food. Bacteria use the alcohol that is produced in wine or beer, and then it produces an acetic acid, which of course will spoil the wine by turning it into vinegar. Pasteur demonstrated that these spoilage bacteria could be killed by heat that was not hot enough to evaporate the alcohol in wine. So he recommended heating the wine enough so that it kills the spoilage bacteria. So this process was called pasteurization. So one of the things that we learned is processes or bacteria were named after the person that discovered it or came up with the process. So pasteurization is the application of a high heat for a short time. And we do this for milk, apple juice. The germ theory of disease. The germ theory of disease says that microorganisms cause disease. In 1835, Agostino Bassi showed that a silkworm disease was caused by a fungus. In 1865, Pasteur believed that another silkworm disease was caused by a protozoan. In the 1840s, Ignaz Semmelweis advocated hand washing to prevent the transmission of puerperal fever from one obstetrical patient to another. So he had a situation where mothers were developing puerperal fever after delivering their babies. And why? Because they weren't washing their hands between patients. So he recommended that they wash their hands in between patients so that they do not, pass it on to another patient. In the 1860s, applying Pasteur's work showing that microbes are in the air and can spoil food and cause animal diseases, Joseph Lister used a chemical disinfectant to prevent surgical wound infections. In 1876, Robert Koch proved that a bacterium causes anthrax and provided the experimental steps called Koch's postulates to prove that a specific microbe causes a specific disease. Vaccination. In 1796, Edward Jenner inoculated a person with cowpox virus, who was then protected from smallpox. Vaccination is derived from vasa, for cow. So that's where they came up with the name. The protection that we get from vaccination is called immunity. The birth of modern chemotherapy. Treatment with chemicals is chemotherapy. Chemotherapeutic agents are used to treat infectious disease, and they can be synthetic drugs or antibiotics. Antibiotics are chemicals produced by bacteria and fungi that inhibit or kill other microbes. The first synthetic drugs. Quinine from tree bark was long used to treat malaria. Paul Ehrlich speculated about a magic bullet that would destroy a pathogen without harming the host. So in 1910, Ehrlich developed a synthetic arsenic drug called salvicin to treat syphilis. In the 1930s, sulfonamides, also called sulfa drugs, were synthesized. The birth of modern chemotherapy. In 1928, Alexander Fleming discovered the first antibiotic. Fleming observed that penicillin fungus made an antibiotic penicillin that killed Staphylococcus aureus, and he observed this by accident. So his plate, he was trying to grow Staph aureus, but what happened was it became contaminated with the mold Penicillium, and when it did, this showed that it inhibited the growth of S. aureus. So from there, in the 1940s, penicillin was tested clinically and then mass produced. Modern developments in microbiology. Bacteriology is the study of bacteria. Mycology is the study of fungi. Parasitology is the study of protozoa and parasitic worms. Virology is the study of viruses. Immunology is the study of immunity. Vaccines and interferons are being investigated to prevent and cure viral diseases. The use of immunology to identify some bacteria according to serotypes was proposed by Rebecca Lansfield in 1933. Recent advances in genomics, the study of an organism's genes, have provided new tools for classifying microorganisms. Microbial genetics is the study of how microbes inherit traits. Recombinant DNA is DNA made from two different sources. In the 1960s, Paul Berg inserted animal DNA into bacterial DNA, and from there the bacteria then produced an animal protein. Recombinant DNA technology or genetic engineering involves microbial genetics and molecular biology. And here are some Nobel Peace Prizes for microbiology research. And you'll see here that we have the TB bacterium, which was discovered by Robert Koch in 1905. Alexander Fleming, remember he discovered penicillin and he got a Nobel Peace Prize along with Chain and Florey. Waxman discovered streptomycin. Prusiner helped to discover prions. Marshall and Warren, they came up with H. pylori and ulcers in 2005. And then in 2008, Sir Houston, he got a Nobel Peace Prize for HPV and cancer, showing that human papillomavirus causes HPV or cervical cancer. And then, of course, Barre, Sanusi, and Montagnier, they were ones that helped discover HIV. Here ends part B of chapter 1.